Welcome, curious minds. Have you ever truly paused to consider the intricate, invisible ballet unfolding within your cells?
It's quite something, isn't it?
The incredibly elegant journey that takes the very blueprint of life, your DNA, and transforms it into a functional part of you. It's anything but a straightforward path, is it?
Oh, far from it. It's a master class in molecular orchestration, really.
Absolutely. So today we're embarking on a deep dive into the fascinating world of RNA metabolism. We're pulling key insights directly from Chapter 26 of Lehninger Principles of Biochemistry, 8th edition.
Right, Nelson and Cox. Good stuff.
And you'll quickly discover that RNA is, well, far more dynamic and ancient than many people realize. It's not just some simple messenger.
No, not at all. It carries genetic information, sure, but it also acts as a catalyst and, amazingly, can even serve as a template for new DNA.
So our journey today is really designed to give you a clear, structured understanding of these complex biochemical pathways.
Exactly. We'll unpack the molecular mechanisms of how RNA is made, explore the extensive processing steps it undergoes.
Discover how RNA can astonishingly serve as a template for other nucleic acids.
Right. Breaking the old dogma.
Right.
And finally, we'll consider the profound implications of RNA's catalytic abilities, which leads us right to the RNA world hypothesis.
Our goal is to provide a comprehensive yet hopefully concise grasp of these core concepts. Perfect if you're looking to quickly and thoroughly understand these fundamental building blocks of life. So let's begin.
Okay, let's start with DNA-dependent synthesis of RNA. We usually call this transcription. It's the very first step in expressing genetic info.
Right, DNA to RNA.
In RNA and DNA, they share similarities, obviously. But RNA has a subtle chemical difference, an extra hydroxyl group on its sugar. And it uses uracil instead of thymine.
Seems like a small change.
It does, but it makes RNA much more flexible structurally speaking. DNA mostly exists as that stable double helix.
The classic picture.
Yeah, whereas most RNAs function as single strands that can fold back on themselves in really complex ways. This allows for incredible structural diversity.
And that structural flexibility is the secret to its unique dual role, isn't it?
Pretty much. RNA is the only macromolecule we know that can both store and transmit genetic information and act as an enzyme, a catalyst.
That dual capacity.
Yeah.
It's really profound. It sparks so much speculation about its role in the origins of life.
Indeed. So we mainly talk about 4 major types of RNA.
Yeah.
Each has a distinct job.
Okay.
Messenger RNAs, mRNAs, they carry the genetic code from DNA to the ribosome.
Protein factories.
Exactly. Then transfer RNAs, tRNAs, act as molecular adapters. They read the mRNA code and bring the right amino acid.
Got it.
Ribosomal RNAs, rRNAs,
Mm-hmm.
are the actual structural and catalytic core of those ribosomes.
So they build the factory and run it, in a way.
You could say that, and then there's this huge, still expanding group called non-coding RNAs, or ncRNAs. They do a massive variety of catalytic, structural, and regulatory things.
It's kind of amazing. You think, okay, only a small bit of DNA codes for proteins, so maybe not much of the genome is active.
Right, that used to be the thinking.
But it turns out, something like 76% of the human genome is actually transcribed into RNA.
It's the current estimate, yeah.
Yeah.
And the vast majority of that transcript is ncRNAs. We're still figuring out what a lot of them do.
So much going on. And this transcription, it's highly selective, right? Not like DNA replication where you copy everything.
Precisely. Only specific genes or groups of genes are transcribed when needed. It's efficient. The cell only makes what it needs when it needs it.
So how does this selective copying happen? What's the machinery?
Well, it's all thanks to RNA polymerase. That's the master synthesizer enzyme.
Okay.
It builds the RNA using a DNA strand as a template. And it uses ribonucleoside, 5-urotriphosphates, ATP, GTP, UTP, CTPs, the raw materials. Needs magnesium ions too.
And it builds in a specific direction.
Always 5-ur to 3-ur for the new RNA strand, which means it reads the DNA template strand in the 3-ur to 5-ur direction. They're complementary.
And unlike DNA polymerase, it doesn't need a primer to get started.
Correct. That's a key difference. It can just start. It also only unwinds a small bit of DNA at a time, maybe 17 base pairs or so in E. coli. That little unwound region is called the transcription bubble.
Bubble.
And inside that bubble, there's a very short, temporary RNA-DNA hybrid, maybe 8 base pairs long. Then the new RNA peels off, and the DNA zips back together behind it.
Clever. But doesn't that unwinding cause problems, like twisting the DNA ahead and behind?
It absolutely does. It creates positive supercoils ahead and negative ones behind. But the cell has enzymes called topoisomerases that act like molecular swivels to relieve that tension.
Keeps everything manageable.
Exactly. Now, in bacteria like E. coli, the RNA polymerase is this complex machine with several parts, or subunits. One crucial one is the sigma subunit.
Sigma factor, I've heard of that.
Yeah, typically 70 in coli.
Yeah,
It acts like a guide. It helps the main core enzyme find the specific starting points on the DNA. These are called promoters.
So it ensures transcription starts in the right place.
Precisely. It prevents wasteful synthesis.
Now, I remember DNA polymerases are super accurate, they proofread. How about RNA polymerase?
Ah, good point. RNA polymerases are, let's say, a bit more error-prone. Their error rate is significantly higher, maybe one mistake every 10,000 to 100,000 bases.
Why is that acceptable? Seems sloppy.
Well, it's less critical, biologically speaking. You make many, many RNA copies from one gene, and most RNAs are eventually degraded anyway. So a mistake in one RNA molecule isn't usually a disaster like a permanent mutation in the DNA blueprint would be.
Ah, okay. Makes sense. Safety in numbers and planned obsolescence.
Something like that. Which brings us to initiation, how it knows where to start. Those promoter sequences we mentioned.
The start signals.
Right. RNA polymerase, guided by sigma, recognizes and binds to these specific DNA sequences. In E. coli, there are consensus sequences, like the NAGASA10 region, often called the TATA box, TATAT, and the nega35 region.
And the enzyme binds there.
It binds first in a closed complex, then it unwinds the DNA locally to form an open complex. Once it starts making the RNA chain and moves past the promoter clearance, the sigma factor usually pops off.
And then elongation just continues.
Yep, another protein called NuSA often binds then, but that initiation step is a major control point for gene expression.
How so?
Well, cells can have different sigma factors that recognize different promoter types. So activating a specific sigma factor can turn on a whole set of genes needed for a particular response, like coping with heat shock.
A coordinated response.
Clever, very. And other proteins, activators or repressors, can bind near promoters to either help or hinder RNA polymerase binding, adding another layer of control.
Okay. And you mentioned in bacteria, transcription and translation can be coupled.
Yes, because there's no nucleus separating the DNA and ribosomes. So ribosomes can actually jump onto the mRNA and start making protein while the RNA is still being transcribed.
Wow. Very different from us eukaryotes.
Totally different. We have compartmentalization transcription in the nucleus, translation out in the cytoplasm.
Okay, so starting is crucial. Controlled. How about stopping? How does it know when the gene ends?
Equally crucial. Termination signals in the DNA tell the polymerase to stop and release the RNA. In E. coli, there are two main types.
Row-dependent and independent.
You got it. Row-independent terminators are pretty neat. The RNA sequence itself folds into a stable hairpin loop structure, immediately followed by a string of U residues.
Like tying a knot.
Kind of. That hairpin structure, plus the weak AU base pairs holding the RNA to the DNA template at that point, destabilizes the whole complex and the RNA dissociates.
And the other type?
Row-dependent termination uses a protein factor called Rho. It's an ATP-powered helicase, like a little motor. It binds to the growing RNA, zooms along it, catches up to the paused polymerase, and somehow helps pull the RNA off.
Okay, that's bacteria. How does it compare in eukaryotes? More complex, I assume.
Oh yeah, a much more complex symphony. We have three main nuclear RNA polymerases. Pol I makes ribosomal RNA precursors, Pol IIa makes tRNAs and some other small RNAs, and the star, for our purposes, is RNA polymerase II, or Pol II.
Pol II makes the messenger RNAs.
Exactly, mRNAs and also many of those non-coding RNAs we mentioned. It's a huge enzyme, like 12 subunits, incredibly complex.
But related to the bacterial one.
Amazingly so. Despite the complexity difference, the core structure and the basic mechanism are remarkably conserved across evolution. Deep roots.
What's special about Pol II?
One really critical feature is its C-terminal domain, or CTD. It's a long, repetitive tail on the largest subunit. Think of it as a flexible scaffold or platform.
What does it do?
It gets modified mostly by phosphorylation and acts as a binding site for tons of other proteins involved in transcription and RNA processing. It coordinates everything.
So Pol II doesn't work alone.
Definitely not. It needs a whole squadron of helper proteins called general transcription factors, or TFIIs, TFIIB, TFIID, TFIHD, and so on.
An orchestra, as you said.
Right. They assemble sequentially at the promoter, along with Pol II, forming a pre-initiation complex, or PIC.
And then what kicks it off?
One of those factors, TFIAH, is key here. It has helicase activity to unwind the DNA at the start site, forming the open complex. And it also has kinase activity that phosphorylates that Pol II CTD tail.
Ah, the coordination hub gets flagged.
Precisely. That phosphorylation is like the signal for ignition and promoter escape. It changes the polymerase's interactions and lets it move forward.
Fascinating. Given how vital these polymerases are, I guess they're good drug targets.
Absolutely. Very useful in research and medicine. For instance, actinomycin D jams itself into the DNA and blocks elongation by any RNA polymerase, bacterial or eukaryotic. Useful lab tool, but toxic.
Okay.
Then there's the rifampin. This one is specific for bacterial RNA polymerase. It blocks promoter clearance, so it stops transcription right at the start.
That sounds like a good antibiotic.
It is a key drug for treating tuberculosis. But bacteria evolve resistance, often to mutations in the polymerase itself, which is a major clinical problem.
Always the arms race.
Always. And then there's otomanitin. This is the toxin from the death cap mushroom.
Nasty stuff.
Extremely potent. It specifically inhibits eukaryotic pol-second very strongly and pull-through to a lesser extent, but not pulsariote or bacterial polymerase. So while it's lethal if ingested, that specificity makes it incredibly valuable for researchers trying to dissect the roles of the different eukaryotic polymerase.
Right. Tool despite the toxicity. Okay, so that's transcription, getting the RNA copy made. But you said it often needs refinement processing.
Yes, especially in eukaryotes. The initial RNA transcript, the primary transcript or pre-mRNA, is often not ready to go. It needs expensive processing additions, deletions, chemical modifications. This profoundly affects where it goes and what it does.
And for eukaryotic mRNA, this processing is quite elaborate, right? Starting with introns and exons.
Exactly. Eukaryotic genes are often mosaics. They have coding segments, the exons, interrupted by non-coding sequences, the introns.
So the whole gene, introns and all, gets transcribed first.
Yes, into pre-mRNA. Then the introns have to be precisely cut out and the exons stitched together. This is called RNA splicing.
And introns can be huge.
Oh, massive. In humans, almost all genes have them. Look at the dystrophin gene. The pre-mRNA is over 2 million nucleotides long, but more than 99% of that is introns that get removed. The final mRNA is much, much smaller.
Wow. Okay, so splicing removes introns. What else happens to mRNA?
Well, very early on, usually while transcription is still happening, the 5-foot end of the eukaryotic mRNA gets a special modification called a 5-foot cap.
Yeah, a cap, like a little hat.
Kind of. It's a chemically modified guanine nucleotide, 7-methylguanosine, linked on backwards via a 5-foot triphosphate bridge. It's weird, but vital.
Why? What's it for?
Several things. It protects the mRNA from being degraded by exonucleases that chew from the 5-foot end. It's essential for transporting the mRNA out of the nucleus. And it's crucial for ribosomes to recognize the mRNA and start translation.
So protection, transport, translation, initiation. Pretty important, Cap.
Very. And its chain is coordinated by that POLTA-CTD we talked about. Even some viruses, like influenza, have figured out how to steal caps from host mRNAs for their own use, cap snatching.
Crafty viruses. Okay, cap at the front, splicing in the middle. What about the end?
Good question. Most eukaryotic mRNAs also get a modification at their 3-bit end, the poly-A tail.
Poly-A, meaning many adenines.
Exactly. After the mRNA is cut at a specific site downstream of the coding sequence, an enzyme called polyadenylate polymerase adds a long string, maybe 30 to over 100 adenine residues, one by one, without a template.
What's the point of the tail?
Like the cap, it helps protect the mRNA from degradation, but from the 3-bit end this time. It also plays a role in translation, initiation, and termination, and nuclear export, again, coordinated via the POL2-CTD complex.
Cap, splice, tail. The eukaryotic mRNA makeover?
Pretty much. Though there's also RNA editing, where the sequence itself can be changed after transcription. Bases added, deleted, or chemically altered. Another layer of complexity.
Okay, let's go back to splicing. You said introns are removed. How?
This is where it gets really fascinating. There are actually different types of introns based on how they're removed. Group I and Group 2 introns are incredible because they're self-splicing.
Self-splicing, they cut themselves out.
Yes. The RNA molecule itself has catalytic activity. It folds into a specific 3D shape that enables it to perform the chemical reactions, transesterifications needed to excise itself and ligate the exons. No protein enzymes needed.
That was a huge discovery, right? RNA as an enzyme or ribozyme.
Monumental. Thomas Suck won the Nobel Prize for it. It showed RNA wasn't just a messenger, it could do chemistry. Group 2 introns even formed this characteristic loop structure called a lariat during the process.
Wow. But most introns aren't self-splicing.
No. The vast majority of introns in our nuclear genes are removed by a huge molecular machine called the spliciosome.
Spliciosome. Sounds complex.
It is. It's made of proteins and several small nuclear RNAs or snRNAs like U1, U2, U4, U5, U6. These snRNAs are packaged with proteins into small nuclear ribonuclear proteins, snRNPs, pronounced SNRPs.
SNRPs.
And these snRNPs assemble on the pre-mRNA. The snRNAs use base pairing to recognize the specific sequences at the intron-exon junctions and an internal branch point site within the intron.
So the RNA parts do the recognizing.
Yes, largely. And here's the kicker. The chemical mechanism the spliceosome uses to remove the intron, forming that same lariat structure, is remarkably similar to the self-splicing group 2 introns.
Suggesting an evolutionary link.
Absolutely. It strongly suggests that the catalytic core of the spliceosome is actually RNA-based, maybe derived from an ancient self-splicing intron, even though it now relies on many protein helpers.
Amazing. Evolution repurposing things.
Constantly. And splicing is often coupled with transcription happening while the RNA is still being made. Afterwards, a protein complex called the exon junction complex, or EJC, gets deposited where introns were removed, marking the mRNA for later quality control and regulation.
So many layers. Now, you mentioned alternative splicing earlier.
Right, this is a huge deal in eukaryotes. The spliceosome doesn't always have to connect exons in the same way. By skipping certain exons or using alternative splice sites, a single gene can generate multiple different mRNA molecules.
Leading to different proteins from one gene.
Exactly. It vastly expands the coding potential of the genome. Same goes for using alternative polyase sites. Using different spots to cut the mRNA and add the tail can change the protein C terminus or affect mRNA regulation.
Can you give an example?
Sure, the calcitonin gene. In the thyroid, it's processed one way to make calcitonin hormone. In the brain, alternative splicing and polyadenylation produce a different protein called CGRP, calcitonin gene-related peptide. Same gene, different products, different functions.
And this has medical relevance. You mentioned SMA.
Yes, spinal muscular atrophy, a devastating disease from a defect in the SMN1 gene. Humans have a backup gene, SMN2, but due to a single nucleotide difference, SMN2 is normally spliced in a way that skips exon 7, producing a mostly non-functional protein. But a drug called nusinersen, which is an antisense oligonucleotide, a small synthetic nucleic acid, binds to the SMN2 pre-mRNA near exon 7. It basically masks signals that cause the exon to be skipped.
So it forces the spliceosome to include exon 7.
Exactly. This leads to the production of enough functional SMN protein from the SMN2 gene to compensate for the defective SMN1, halting the neurodegeneration. It's a prime example of targeting RNA processing therapeutically. Incredible breakthrough.
Truly amazing. Okay, so mRNA processing is complex. What about other RNAs like rRNA and tRNA?
They get processed too from longer precursors. Ribosomal RNAs in bacteria are often transcribed as one long unit and then cleaved by specific nucleases. In eukaryotes, it's more complex, happens in the nucleolus, guided by other small RNAs called snoRNAs within snorin P complexes.
More RNA guiding RNA processing.
It's a theme. And tRNAs also start as longer precursors. Bits are cleaved off both ends. One enzyme involved, RNase P, is another classic ribozyme. Its RNA component is essential for catalysis.
Another RNA enzyme.
Yep.
Yeah.
Plus, tRNAs get the sequence CCA added to their 3-foot end. That's where the amino acid attaches. And many of their bases undergo extensive chemical modifications, which are critical for their structure and function.
And those microRNA.
Myrinase. These are tiny regulators, about 22 nucleotides long. They start as much longer primary transcripts, get processed in the nucleus by an enzyme called DROSHA, then exported and processed again in the cytoplasm by dicer.
Double processing.
Right. The mature myRNA then gets loaded into a protein complex called RISC, RNA-induced silencing complex. This complex then uses the myRNA as a guide to find target mRNAs, usually binding to their 3-foot untranslated regions.
And then what?
Depending on the match, RISC can either trigger the mRNA to be degraded or just block its translation into protein. It's a major way genes are regulated post-transcriptionally.
So much control after the gene is transcribed.
Absolutely. And finally, RNA doesn't last forever. Regulated degradation is just as important as synthesis for controlling gene expression levels.
How does that work?
The pathways differ between bacteria and eukaryotes, but generally involve removing protective features like the cap and tail, followed by digestion by nucleuses. In eukaryotes, a major player is the exosome, a large complex that degrades many types of RNA from the 3'.
Like a cellular recycling center for RNA?
Sort of, yeah. Keeps things tidy and allows expression levels to change quickly.
Okay, this is intricate. But so far, it's all been DNA template, RNA, maybe protein. You mentioned breaking the dogma.
Yes. Now we venture into territory where RNA itself serves as the template. This was a huge shift in thinking, driven largely by studying viruses.
You mean RNA making DNA?
Precisely. Enter reverse transcriptase. This is an RNA-dependent DNA polymerase. It reads an RNA template and synthesizes a complementary DNA strand.
Found in.
Famously discovered in retroviruses like HIV, they have an RNA genome, but when they infect a cell, they use reverse transcriptase to make a DNA copy of their genome.
And that DNA copy then gets into the host's DNA.
Exactly. It integrates into the host chromosome, becoming a provirus. Then the host cell's own machinery transcribes it back into viral RNA and proteins. The discovery by Temin and Baltimore was revolutionary information flowing from RNA to DNA.
Shaking the foundations. What else does reverse transcriptase do?
It's actually multifunctional. It makes the first DNA strand using RNA template, then it degrades the original RNA template. It has RNase H activity. And finally, it synthesizes the second DNA strand using the first DNA strand as a template. It makes a DNA double helix from an RNA single strand.
Wow. And it's accurate.
Not very, like RNA polymerases, but often even more so, reverse transcriptases lack good proofreading. HIV's reverse transcriptase is notoriously error-prone.
Which helps the virus evolve quickly, dodging immune systems and drugs.
Exactly. That high mutation rate is a major reason why HIV is so challenging, and why combination drug therapy targeting different viral enzymes, including reverse transcriptase, is essential.
Makes sense. Are there connections to other things in our genome?
Fascinating connections. Eukaryotes have mobile genetic elements called retrotransposons. They look a lot like retroviruses, structurally, and they move around the genome by making an RNA copy of themselves, then using reverse transcriptase to make a DNA copy that inserts somewhere else.
Like molecular parasites within our DNA?
Pretty much. And even some of those self-splicing introns, particularly group 2 introns, can move around. Some encode proteins with reverse transcriptase activity, allowing them to spread via an RNA intermediate, a process called retrohoming. It suggests introns and retroviruses might share ancient origins.
Mind-boggling evolutionary echoes. Is reverse transcriptase only in viruses and transposons?
No, we have a crucial one in our own cells. Telomerase.
Telomerase. So that's involved in chromosome ends, right? Telomeres.
Yes. Linear chromosomes have a problem. The normal DNA replication machinery can't fully copy the very ends, so chromosomes will get shorter with each cell division.
Which would be bad.
Very bad. Telomeres are repetitive DNA sequences at the ends that act as protective caps. Telomerase is a specialized reverse transcriptase that maintains telomere length.
How does it do that?
Telomerase is actually a ribonuclear protein. It contains its own RNA molecule that serves as the template for synthesizing the repetitive telomere DNA sequence. It extends the 3' end of the chromosome, allowing the replication machinery to then fill things in better.
So it uses an RNA template to make DNA, just like retroviruses, but for a totally normal, essential cellular function.
Exactly. And telomere length and telomerase activity are linked to cellular aging and cancer. In most of our body cells, telomerase activity is low, and telomeres shorten over time, contributing to senescence. Cancer cells often reactivate telomerase to achieve immortality.
A double-edged sword. Okay, so RNA can make DNA. Can RNA make RNA?
Yes, indeed. There are RNA-dependent RNA polymerases, often called RNA replicases. These are found primarily in RNA viruses that don't use reverse transcriptase, like influenza, polio, and coronaviruses.
So they just copy their RNA genome directly into more RNA.
Right. They use the viral RNA as a template to make complementary RNA strands, which then serve as templates to make more copies of the original genome. Like reverse transcriptases, they generally lack proofreading, contributing to rapid viral evolution.
Makes sense for viruses. Do eukaryotes use them?
Some do, yes. Plants, fungi, some protists use them, often involved in processes like RNA interference, making small interfering RNAs, cerminase.
So RNA-dependent synthesis is pretty widespread, just maybe less central than DNA-dependent synthesis in cellular life.
That's a good way to put it. And what's striking is the structural similarity. Reverse transcriptases, telomerase, viral RNA replicases. They often share a common protein fold, suggesting a deep evolutionary kinship.
Which brings us back to RNA's catalytic abilities, the ribozymes.
Yes, one of biochemistry's biggest surprises. We've mentioned self-splicing introns, RNase P. These RNAs aren't just passively folding, they're actively catalyzing chemical reactions.
And they act like real enzymes.
Absolutely. They have specific 3D structures essential for function. They dramatically accelerate reaction rates. The tetrahemina intron speeds up hydrolysis by 10 times. They show saturation kinetics, column to PM, can be competitively inhibited, are highly specific, often using guide sequences within the RNA itself.
Can they do multiple reactions like a protein enzyme?
Some can, yes. They use similar catalytic strategies to orienting substrates, sometimes forming covalent intermediates using metal ions. And remember, the spliceosome's core is likely RNA, and the ribosome itself uses RNA to catalyze peptide bond formation.
The very heart of protein synthesis is an RNA machine.
Precisely. RNA's catalytic role is ancient and fundamental.
Which leads directly to the RNA world hypothesis.
Exactly. Proposed by Woese, Crick, Orgel, and others. The idea is that early life might have relied on RNA for both information storage, like DNA does now, and catalysis, like proteins do now. An RNA-based world preceding the DNA-protein world we know.
What's the evidence for this radical idea?
Several compelling lines. First, prebiotic chemistry experiments show RNA bases like adenine can form spontaneously under plausible early Earth conditions. And many modern enzyme cofactors like AGP, NAD, FAD, coenzyme A still contain adenosine, an RNA component. Relics, perhaps.
Interesting. What else?
Second, the existence of ribozymes themselves. If RNA can catalyze reactions, including potentially its own replication, then an RNA-based life form becomes conceivable.
Makes sense. Proof of principle.
Third, the expanding catalytic repertoire. Using techniques like SALEX, scientists can artificially evolve RNA molecules in the lab to catalyze all sorts of reactions or bind specific targets. These are called uptamers. It shows RNA has the potential for diverse catalysis needed for primitive metabolism.
But RNA is chemically versatile.
Weirdly. Fourth, the ribosome, as we just discussed. Protein synthesis being catalyzed by rRNA is powerful evidence for RNA's early central role.
Right, the machine that builds protein builders is made of RNA.
Fifth, extant vestiges. Things like retroviruses and retrotransposons operating on RNA intermediates could be seen as molecular fossils. Glimpses into ancient RNA-based strategies for replication and movement.
Echoes from the past.
And sixth, exciting progress in the lab. Researchers like Gerald Joyce and Philip Holliger have engineered ribozymes that can catalyze the formation of other RNA molecules, even acting like primitive RNA polymerases, or cross-catalyzing each other's synthesis, demonstrating RNA's potential for self-replication and evolution.
Getting closer to making RNA replicate itself in a test tube.
They're making amazing strides. It really bolsters the plausibility of the RNA world.
So wrapping this all up, what's the big takeaway for listeners?
I think it's the incredible versatility of RNA. It's not just a messenger. It's transcribed, processed in intricate ways, can act as a template for DNA synthesis, and has this ancient fundamental catalytic ability that likely predates protein enzymes.
It really shows that RNA is this dynamic, ancient, and still evolving player. Understanding its metabolism isn't just academic. It's about understanding life's operating system, its vulnerabilities like in viruses or genetic diseases, and its amazing adaptability.
Absolutely. And thinking about the elegance of RNA's roles, especially catalysis and information transfer, it makes you wonder, what's the next big aha moment going to be? Will we discover new forms of genetic catalysts? Or figure out how to engineer RNA for applications we can barely dream of now?
So many possibilities still hidden in this incredible molecule. Well, thank you for joining us on this really deep dive into the world of RNA.
My pleasure. It's a fascinating topic.
And thank you, our listeners, for being part of the Last Minute Lecture family.